So anyways, this is a novel coronavirus from patients with pneumonia in China 2019. This is from Nazu, and then I'm not going to pronounce these names. And this is from the China Novel Coronavirus Investigating and Research Team. All right, now let's take a look at the paper abstract or summary. I'm just used to the word abstract. So let's go over the summary or abstract of the paper. And here's the first part. Then I'm going to go over some of the technical terms and then continue back on the paper. So summary: in December 2019, a cluster of patients with pneumonia of unknown cause was linked to a seafood wholesale market in Wuhan, China. A previously unknown beta coronavirus was discovered through the use of unbiased sequencing in samples from patients with pneumonia. All right, so now let's go over some of these terms. So unbiased sequencing refers to genetic sequencing techniques aimed at minimizing any biases or errors in sequencing and data processing. And pneumonia is inflammation of the lungs, primarily the alveoli, and is supposedly caused by, quote, infection with, quote, viruses or bacteria, or less commonly by other microorganisms. And again, just a note here. So this one here, I have links to all these technical terms. This one I used ChatGPT. It's just because the Wikipedia didn't have a complete information on that. And then here's pneumonia. This was from Wikipedia and so on. And I'll just highlight these. All of these links are all on there. All right, continuing further. So inflammation is a complex biological response to harmful stimuli and is characterized by heat, pain, redness, swelling, and loss of function. And these are just mainstream explanations of these. And alveoli are millions of tiny air sacs in the lungs where oxygen/CO2 gas exchange occurs with the outer environment and the blood, and back and forth and so on. And in other words, that's pretty much where the source of breathing comes in, or getting the oxygen from the atmosphere and then exhaling the CO2 waste out of the body. And yeah, so here's this is the trachea or windpipe, and then you have the lungs, and then inside these lungs, you have it just branches out over to these bronchi, and then these smaller bronchi, and then or bronchioles, and then they expand outwards to these alveoli, that are these millions of air sacs, and so on, and you connect them to these blood vessels and the outer atmosphere connecting to them. All right, so inflammation of these alveoli inside the lungs is basically considered pneumonia. So they get inflamed, there's a lot of heat, pain, stress, redness, swelling, and so on. And so now let's continue further. So those were the terms there. Continuing further, or actually before I continue further, I just want to add another definition here for beta coronavirus. I'll put that right here. So beta coronaviruses are one of 4 genera or classes of coronaviruses, and includes SARS-CoV-2, the alleged, quote, virus that, quote, causes COVID-19. So that's basically a classification of these so-called viruses. Anyways, going further. So continuing further in the paper, in the summary, human airway epithelial cells were used to isolate a novel coronavirus named 2019 nCoV, which formed a clade within the subgenus Sarbecovirus, and then this is the inside the Orthocoronavirinae subfamily. All right, so different from both MERS-CoV and SARS-CoV, the 2019 nCoV is the 7th member of the family of coronaviruses that infect humans. And enhanced surveillance and further investigation are ongoing. And this paper was funded by the National Key Research and Development Program of China and the National Major Project for Control and Prevention of Infectious Disease in China. Interesting. All right, so now let's continue into more of these technical terms. So epithelial cells right here. So epithelial cells are the cells that line the outer surfaces of organs and blood vessels And the human airways, so this is human airway, nose, mouth, and down to the lungs. These ones have the pseudostratified type of these epithelial cells. And pseudostratified is named such that since they have the appearance of consisting of multiple layers of cells, but are actually just one layer. And here's types of epithelium. So you have the simple squamous, looks like that. Simple cuboidal, looks just like cubes like that. And then simple columnar, so just like columns. And there's a transitional, it's like columns and cuboidal. There's stratified cuboidal or cuboidal, and then there's stratified squamous. Kind of looks like flat and stratified as well. And then you have the pseudo-stratified columnar or columnar. So this one's a columns, but then this one looks like it's small. It's stratified with multiple layers, but they're actually just all squished like sideways like that. So they're not actually on top of each other. So yes, that's a pseudo-stratified, and these are the ones that line the nose, mouth, and down to the lungs. And and so here's a graphic of that pseudo-stratified ciliated columnar epithelium. So there's lots of terms. There's a lining of this throat right there, or the trachea. And then there's the cilia right there. These are fibers, hair-like fibers, and they move up and down the mucus and particles in a coordinated fashion to get them out of the mouth and so on. And there's the nuclei, there's the cells, and there's the cytoplasm, which is all the material inside the cell. And there's a basement membrane, there's a lamina propria, or loose connective tissue, on this side of these organs right there. It's fascinating stuff. And going further, so this forms a clade, or clade, I'll call it clade. And this is just a grouping of organisms that have a common ancestor and contains all of its descendants. For example, the blue and orange groupings are clades, but the green is not a clade. So in other words, this blue contains all of its descendants, so that is a clade. And likewise, the red also contains all of the descendants, that's called a clade. But the green is not, because there is this part right here. It does not contain this segment here. But then again, you can make the blue and green, that'll be a clade. Or you can make this entire thing, and that'll be a clade as well, because they'll contain all the ancestors. So again, that's just a taxonomical grouping setup. In the subgenus, and this is from this part right here. So these they isolated a new or novel or new coronavirus, forms a clade within the subgenus Sarba-coronavirus and this subfamily and so on. So the subgenus, that is so a subgenus is a taxonomical or grouping directly below genus. And for example, the genus Panthera has subgenera, just plural of subgenus, the lion, tiger, jaguar, and leopard. So yes, these are Panthera or Panthera genus, and these are subgenus. Anyways, then the Sarbecovirus, that's short for SARS beta coronavirus, is a subgenus of beta coronavirus, and beta coronavirus is one of 4 genre or plural of genus of coronaviruses. The others are alpha, gamma, delta, and so on. And then orthocoronavirinae is a subfamily in the family of Coronaviridae. And then the term coronavirus refers to any member of the Coronavirinae subfamily. Anyways, that's just a lot of technical taxonomical terms. And just to give you just an overview of how this setup is. So if you have a domain, for example, eukaryotes or multi-celled organisms, and you have a kingdom, for example, animals or Animalia, and then you have this phylum, it's Chordata with backbones, class Mammalia, mammals, and then you have order Carnivora or carnivores. You have the family Canidae or canines. You have genus Vulpes, species, and that is Vulpes. Same thing as red fox. So that's just a taxonomical setup. So now for the coronavirus, the SARS-CoV-2, there's a taxonomical rank for it. So you have unranked is just as virus. Unranked is interesting. And then you have the realm. That's a new one I've heard before. Ribaviria. And kingdom Orthoranviri. Anyways, there's phylum. You have this, and then class, and so in order. Then you have the family is a Coronavirus, Viridae. Then you have genus Beta coronavirus, and then subgenus Sarbecovirus. And then you have the species, which is back to, this would be like the red fox. Now you have the species severe acute respiratory syndrome related viruses, and then the actual virus, severe acute respiratory syndrome coronavirus 2. So a whole bunch of these species there, all related. All right, so that was the abstract. Now let's just summarize it quickly. Again, the main idea of this paper, or this video that I'm reviewing, is going to be on the virus isolation, but I wanted to go through the entire paper in detail first. So summary of the abstract. Let's go over a quick summary of the abstract. # summary of the summary. So patients with pneumonia or inflammation of the lungs of unknown cause was, quote, linked to a seafood market in Wuhan, China in December 2019. And then genetic sequencing of samples from pneumonia patients was used to, quote, discover an unknown coronavirus. And then human airway epithelial cells, or cells that line the nose, mouth, and down to the lungs, were used to, quote, isolate a novel or new coronavirus named 2019 nCoV. All right, so to break it down even further, so basically they found some patients with inflammation of the lungs, then they did genetic sequencing to discover a new virus, and then they somehow isolated it using the cells that line the nose, mouth, and lungs, and so on. So yes, very interesting. So let's we're going to look through the methods and so on to see if this all makes sense.